Hi, I'm Rhys and I use genomics to analyse biological data. We investigated multi-drug resistant Escherichia coli sequence type 131, which disproportionately causes bacteremia in whales. Despite the significant morbidity and mortality associated with E. coli bacteremia, strains responsible for this increase in whales are poorly characterised. ECB has become a major concern in Wales, with the incidence of infection increasing. In 2017, at least 1 in 1,000 individuals had these infections. Over 2013 and 2014, microbiology labs submitted all non-duplicate E. coli blood isolates to Public Health Wales for genome sequencing. Our genetic data represents a collection of ST131 gathered from this national surveillance. After stringent quality control using bioinformatics tools, we analysed more than 140 strains collected from 20 hospitals. By combining our Welsh strains with strains sequenced elsewhere, it's apparent that fluoroquinolone-resistant clade C is predominant. This resolution implies that the increase in ECB in Wales is likely a population-level increase rather than that of a single strain. To identify any evidence of phylogeography, we avoided recombinogenic artifacts by focusing on clade C. A unique subclade was identified in North Wales. This lineage is within the C2 subclade, which is associated with resistance to broad-spectrum antibiotics like fluoroquinolones or third-generation cephalosporins. Briefly, this identified sublineage differs by 2 SNPs in genes relating to fitness or virulence. These strains have also lost the capsular biosynthesis genes which encode the K5 antigen. Local transmission within North Wales is likely due to low genomic diversity between these strains, which have a median pairwise distance of 48 SNPs. Our temporal analyses show that the common ancestor to this identified sublineage diverged from that shared with a Canadian strain between 1998 and 1999. These strains have persisted in Wales since the early 2000s and have continued to cause infections years later. It is likely that this strain is an endemic clone that is well adapted to the environment. While it's tempting to speculate a North American origin, it's important to recognise that instead, this points towards the global dispersion of ST131. Overall, we describe the first in-depth genomic epidemiology of significant E. coli strains contributing to bacteremia in Wales. The identification of a novel lineage within North Wales highlights the value of genome sequencing and the ability to determine the relatedness between strains that typically isn't offered with traditional typing techniques. By gaining a detailed understanding of E. coli strains in circulation, it is possible to develop and improve public health measures to reduce the risk of bacteremia and battle escalating antimicrobial resistance. Thank you.